Hi, everyone. I'm Heather Patterson. I'm one of the technical service managers at Control Solutions, Inc. Control Solutions, or CSI, we are a pesticide manufacturer based out of Pasadena, Texas. And our team here is dedicated and driven to developing and delivering an innovative solutions for you guys to use for a wide range of pest problems that you face in the field, including termites. Now, in today's presentation, we're not going to really be talking about the different termite products or formulations that we make. But we'll be focusing on the 12 strange, fascinating, and useful termite facts that PMPs should know. If you do have any questions about our products, feel free to reach out to Heritage or myself or somebody else from CSI, and we would be more than happy to help you with those questions. I want to thank Heritage Supply for inviting CSI to be part of their virtual academy this year. And if you do have any questions, also feel free to pop those in the chat, and we'll try to get those answered for you as quickly as possible. All right, without further ado, quick look at our outline. I have a total of 12 strange, fascinating, and useful termite facts that I'll share with you today. And it's going to be touching on all the different termite topics. We're going to discuss a little bit about social behavior, different organisms that live in close association with termites, discuss a little bit about their morphology, molting, caste systems, how castes are determined inside of a termite colony, as well as foraging and feeding from a control standpoint, and others. So as I share these various termite related facts, most will have a slide prompting this question. How does this information help me? How does it help the PMP? How can we use this information that we just learned to help us be better at or more effective at our jobs? How does this information help us to understand termites and the various tools and formulations that we have available to control them? And so after we go through each of these facts, we'll take a moment to kind of think about this question, and I'll share what I think is a key takeaway, but I think it's awesome if you come up with a different conclusion. I think this is a good opportunity to also mention the difference between basic science and applied science, because you'll notice as we go through these various facts that a lot of these kind of fit that basic science category. And ultimately, it's based on purpose or utility. A research approach or scientific question can either be basic or applied in nature. And so specific to our field of interest, urban and structural pest management, basic science seeks to expand our knowledge and broaden our understanding of the various pests that we face. We want to know about the biology and behavior and preferences of bed bugs and mosquitoes and termites and ants. So to contrast this, applied research and science is more focused on discovering practical solutions to very specific problems. And in many ways, applied science depends on the findings that we get from basic research. So boiling it down, basic science seeks to more so know thy enemy, know the pest, whereas applied science works to use that information to find ways to develop tools to conquer it, to control it. So let's jump into the first fact that we have lined up for you today, and that is termites have been around for a long time. I know this is not a very surprising fact for most of us here. We're very well aware that the termite fossil record is quite old, but hopefully after we go through this section, we have a newfound appreciation for just how long these organisms have been around for, and what that means for management. There's a lot going on here. I know the first time I looked at this slide, I'm like, what do I focus on? What do I pay attention to? And I'll help break it down. We'll digest it a little bit together so that it can be a little bit more useful to answer the ultimate question that we have. So what we're looking at here is what's called a condensed geological timescale, and present day is at the very top. So today at the very top of this timescale, we are living in the Phanerozoic eon of the Cenozoic era. And so scientists consider anything in this Cenozoic era, which is from the top all the way down to that little circle with the 66, circled 66, that's the Cenozoic era. Everything from now all the way back to 66 million years ago is considered recent life. And as we work our way down along this time scale, it goes beyond 500 million years ago. You're going to recognize some different time periods and eras, and also see the emergence of animal groups that you're quite familiar with. If you look at that circle with the 66, that's separating the end of the Cretaceous period, that green box, from the peach box above it, the Paleogene. and you scroll all the way to the right, there's a little peak there. That peak represents a mass extinction event that ultimately killed off several animal groups, including famously the dinosaurs. And you see that dinosaur group kind of towards the right side of this scale here, and you see where it cuts off at that 66 line. Because of the mass extinction event that happened at that time period. And so that's what each of these little white blobs on this time scale represents, is an animal group. And the wider the area of that white blob, the more speciose, the more abundant that group of animal is, and the more narrow it is, the fewer species there are. So you'll see that for the animal groups represented, there's a total of 7 that reach the very top of this time scale. That means those are the 7 groups that are still in existence today. So starting out on the group at the left, those are called brachiopods, and you can see they've been around for 500 plus million years. Fossil records indicate there at one period there was over 300 or 30,000 different species, and today there's less than 400. That's why you kind of see that tapering towards the top of the time scale here. To the right of that, we have the group called echinoderms. You're very familiar with these: sea urchins, starfish, sea cucumbers. If you go all the way down to where it originates, 450 million years ago in the Ordovician time period, as time has gone on, that white area, you can see it's expanding. That indicates that it's become a more successful group. More species have joined in that echinoderm animal group. From there, you have nautiloids and agnatha, very, very thin line at the top here. These have been around for hundreds of millions of years, but only very few species are still in existence today. And then at the end, these are going to be animal groups that you're very familiar with: fish, birds, and mammals. And you can see the birds and mammals in particular are going to be the youngest of the animal groups shown here. Okay, now that we know what the heck we're looking at, let's go back to our original question, which is, where do termites show up in this time scale? You've probably noticed that this illustration lacks insects altogether. And what we do know about these organisms, they are very old, but specifically, we can expect termites to show up around here. The Jurassic period of the Mesozoic era, and the Jurassic period began somewhere around 200 million years ago, characterized by warm, wet climate, and this climate gave rise to lush vegetation and abundant life. And you can see kind of here the animal groups are doing very well, very speciose and abundant during this period. This is when Earth's climate turned essentially into perfect termite weather, shifting from hot and dry to now humid and subtropical. So the evolutionary history of termites, it intrigues biologists and scientists for a number of reasons, including their eusocial behavior, caste diversification, also their ecological importance. I know, when we talk about beneficial organisms, termites don't really come to the forefront of our minds. We think pollinators and bees and butterflies and ladybugs, even spiders, but termites are also very important beneficial organisms because they play such an important role in decomposing organic matter, wood, and the environment. In addition to that, we know that as they're tunneling in the soil, they're aerating the soil, they're reducing compaction, which helps plant growth. The plant roots and areas where termite populations are found, they're able to more effectively take up water and other nutrients from the soil because it's less compacted and aerated from termite activity. So yes, termites are very much a beneficial organism in that respect. Of course, the ones we're concerned about are structural pests. But back to fossils, they're going to be key to our understanding of the history of termites because in many ways, fossils are only direct evidence of the past. Now, of course, like with other insect fossil records, termite fossil records are quite fragmentary in number, quality, as well as the type of preserved specimens or remains that we have. Interestingly, many of the species are going to be known only from a single preserved termite specimen, and nearly all of these termite fossil specimens are of the alate form, like what we see here. What we're looking at is an image from a publication in 2009 titled Description of an Early Cretaceous Termite. We saw the Cretaceous period on the geological time scale a few slides ago. and its associated intestinal protozoa with comments on their coevolution. Now, I know many of you have learned early on in your pest management career how to differentiate ants from termites. And so, for those of you who are new to pest management, a couple of things that we know to look for include one, the antennae. So you can see this specimen very well has this bead-like antennae. It's known as maniliform antennae. It looks like a string of beads. That's indicative of a termite. Ants gonna have these geniculate antennae that kind of look like they're elbowed. Another thing that we look for when we're comparing ant versus termite alates are the wings. Termite alates are going to have a forewing and a hindwing of equal lengths, whereas an ant alate, the forewing and hindwing will be of different lengths. And then also we can look at the body shape. Termites are going to have a more chunky body shape, whereas we know ants have that thin waist or petiole that attaches the gaster to the thorax. So those are the three things that help us differentiate one another. You can see clearly in this image here, we're looking at a termite alate, and really the dead giveaway is the antennae. Uh, but the wings are kind of crumpled up here, but again, the wings would fold nicely over one another and be the same length. So what is the point that I want to make with this particular image? Well, it's interesting because this is a fossil specimen of a termite preserved in a piece of Burmese amber. So think Dr. John Hammond from Jurassic Park, right? You know, he had that piece of amber with the mosquito in it. And that is a wonderful way that we've been able to look at insects from millions of years ago or in these pieces of preserved amber. For this particular fossil specimen, you can see the abdomen's kind of broken or ruptured at the end. And scientists can take this piece of amber and stick it under the microscope, and inside the gut of the termite, they're able to see those diverse protists, the gut microbes that we know that termites depend on in order to help them effectively digest cellulose, which is a very complex, difficult sugar for many animals and organisms to break down, but we know that termites do that very, very well. And so those gut microbes, the scientists were able to see were in the gut of this termite, but not only that, they're kind of floating around that broken part of its abdomen here in the amber matrix. So this is fossil evidence of that symbiotic relationship between termites and gut microorganisms from millions of years ago. So termites have colonized many different habitats and climatic zones. They're going to be some of the most abundant animals, especially in tropical ecosystems. The timing and the rise of abundance that we see in termites, as well as their dispersal events that led to modern lineages, it's not very well or entirely understood. But what we do know is that termites are very, very old, very, very successful organisms. They've survived millions of years of planetary changes. They're hardy little buggers, and they weren't taken out by those extinction events that we saw a few slides ago. They're not going anywhere anytime soon. Okay, fact number 2, termites have undertakers. How do we define undertaker? I'm not talking about the WWE Hall of Famer from Houston, Texas. Undertakers, of course, are ones whose business it is to prepare the dead for burial. They're the ones arranging and managing funerals. So why do termites have this? Why do they do this? Why do they manage their dead? Well, first, as we know, the sanitary issues and concerns associated with exposure to corpses, um, it's going to be universal, right? It's especially concerning among social organisms that are living in these enclosed nests with dense populations. Their colony structure can make them very vulnerable to unhealthy organisms, contagious pathogens, parasites, etc. So to maintain healthy colonies, social insects like termites, like ants, as well as some species of bees and wasps, they've evolved some pretty sophisticated mechanisms to counter the threat of disease, both at the individual and the colony level. When we talk about social insects, there's 3 criteria that need to be met in order to be classified and considered a social insect. The 3 criteria include reproductive division of labor. So there's obviously the queen or the reproductive individual in the colony who's responsible for population growth and egg laying. And then you have your non-reproductive individuals that are taking care of other tasks in the nest, like foraging for food, defending the nest, taking care of the queen and the brood. So that's criteria number one. Criteria number two is overlapping generations. So you have old individuals in the colony that are on the brink of death, and then you have new eggs being laid every day, and then everything in between. So overlapping generation. And then the third criteria is cooperative brood care. Everybody's invested in pitching in to help take care of the eggs, the youngsters in the colony. Everybody's invested in taking care of those eggs that they may not have even laid, right? They're related to them, but they did not lay them, and that's kind of unique to these social insects. And so that's why termites and ants fit that criteria, and again, some species of bees and wasps. And so again, they have these mechanisms to help counter the threat of disease. These include things like active immune responses. There's some examples of behavioral adaptations like corpse management, this undertaking behavior, which happens to be one of the more intriguing behaviors observed in social insects. And it's not only important, but necessary for the overall well-being, survival, health, and protection of the nest. There were some notable findings that came from this study, like which variables affected undertaking behavior. Some factors that affected how termite individuals were handled included a couple of things, like one, the length of time that the dead termite was dead for, right? Was it freshly dead, or had it been dead for quite some time and it was starting to get a little funky? And then two, which caste the individual belonged to. Was it a worker? Was it a soldier, a reproductive member of the nest? So these are things that determined how it was handled. So what happened with the freshly dead individuals? Well, for the study that we're referencing here, freshly dead was described as an individual that had been dead for less than 64 hours. What would happen to freshly dead individuals? Well, they were collected from the test arenas by other nestmates to be brought back to the colony and cannibalized. Every time, didn't matter what caste they belonged to, they were eaten. So why do termites partake in cannibalism of their dead relatives? Well, for a few reasons. Number one, it's nutrient recycling. They are a source of moisture, nutrients, and also point two, they're a source of those useful gut microbes that, again, they depend on to help digest cellulose. Now, we know that termites engage in trophallaxis to help share those gut microbes. Same is true. They are going to take advantage of obtaining some of those gut microbes through cannibalism as well. Now, feeding on dead colony members can replenish those symbionts in the digestive system. And in addition to that, practicing cannibalism can help prevent the development of potentially dangerous or lethal pathogens. For example, growths that may be found on dead bodies that can harm the health of the nest. What we're looking at here is a figure from this publication that illustrates the design of the test as well as measured endpoints. We're evaluating the live termite workers' response to freshly dead corpses of different castes. So the top image shows detection time, which is how long it took the live worker to find the dead termite in the testing dish. And the bottom image shows the retrieval time, for how long it took that live worker to bring the dead termite out of the testing dish back through the connection tube to the colony on the other side. Here are the results. So we have detection time on top in graph B and the duration of retrieval time at the bottom, graph C. Starting at the top, the blue bar represents the freshly dead worker, orange is freshly dead soldier, gray is freshly dead short-winged bud nymph, abbreviated SWBN, and then long-winged bud nymph is the black bar here as well. So you can see for the freshly dead worker and soldier, it took about 10 minutes for that live worker to find them, and slightly longer for the nymphs, with the long-winged bud nymph taking closer to 20 to 25. When you're talking about the duration of retrieval, again, very similar for worker and dead soldier, somewhere around 20 to 25 minutes, and then significantly longer for the nymphs, especially the short-winged bud nymph, which took closer to 50 to 55 minutes to retrieve. Now, again, regardless of detection and retrieval time, all individuals and all castes were cannibalized. So the question here is, well, what happened to the not so fresh bodies? Again, these individuals had been dead for greater than 64 hours, and when they were found in that test arena, one of three things was observed. The live worker would either, one, bury the dead termite on site in the place where it was found in the testing dish. Two, they observed that dead individual being brought back into the nest on the other side to be buried there. And three, they also observed this building of a wall or barrier between the dead individual and the colony. So why would they do this? Why would they take the time to bury these individuals or construct a barrier? Well, it's likely to separate these decomposing bodies from the colony and also help to prevent or reduce exposure to disease, fungus, and other pathogens. So this is another figure from that publication that illustrates those three different behavioral responses to, again, the not so fresh dead bodies. The top image shows that burial in the testing dish behavior. The middle one, that's showing the live worker walling off the colony from the dead soldier. And then at the bottom here, we have an image that shows that live worker retrieving that dead soldier to bring them through the connection tube to be buried on the other side in the living colony. So all three of these behaviors were observed, but not equally. So here are the results shown here. The y-axis or the vertical axis is those not so fresh dead bodies. We have long wing bud nymph at the top, followed by short wing bud nymph, soldier, and worker. And you can see black dominates this graph here. That means for every time a not so fresh nymph was found, as well as the worker, the live worker would bury it inside that testing dish. But not the case for the soldier, right? One of those three things happened, and if the soldier was found, about 50% of the time it was buried in the testing dish. About 40% of the time, the live worker would create that wall of separation between the testing dish and the living arena. And then about 10% of the time, it was moved and then buried in the living colony, opposite of the connection tube. So the real question here is, why do we care about this behavior? Why do we care about undertaking behavior in a termite colony? Well, most of the liquid termiticides that we use in this industry, they're non-repellent formulations, right? Think fipronil, think imidacloprid. There's several examples we could talk through. Typically, these non-repellent formulations are going to be slower acting as well. Baits, they're also non-repellent. And so that means these are compounds that are not going to be detectable by termites. And so the termites that die from exposure to these products are going to be handled in some fashion. And in many cases, as we learned here, if they're freshly dead, they're going to be cannibalized. So the handling of dead termites, even if they're not so freshly dead, they're going to be buried. This behavior is going to increase the exposure of healthy termite nestmates to the termiticide. And this ends up being another mechanism that active ingredient might be moved through the termite colony. They're helping us move those particles through the nest. Coming along to fact number three. Fact number three is termites have unique forms of defense. When I mention termite colony defense, we're all probably picturing the same thing in our minds. That's the termite soldier, right? Specifically the ones that have the pinchers. And that is true for some species, especially the ones we're concerned about in structural pest management. But we're going to take a look at some other more unusual forms of termite defense that you may be less familiar with. Now, I do want to start off with the obvious type that I just mentioned, these powerful mandibles that the termite soldier uses to crush or pierce their enemies. These mandibles, as we know, are very useful for determining what type of termite we're dealing with. They're really important for identification. In this image, you can see that these are both subterranean termite soldiers. They have those long curved mandibles that are smooth, that kind of cross at the tip here. They lack teeth and serration. So on the left here, we're looking at a soldier that has this seed-shaped head or round head. That is the Formosan subterranean termite soldier. And on the right, that boxy rectangular head belongs to the Eastern subterranean termite soldier. If we were looking at the mandibles of a drywood termite soldier, for instance, Incisothermes minor, we would see that their mandibles do have teeth and serrations on theirs. There's going to be some variations too, of course, but this is, again, the form for many of the species that we're concerned about in structural pest management. Here's another form of colony defense. This is a termite soldier that is a drywood termite that's found in Cameroon, and it looks like these would be pinching, crushing mandibles, but in fact, this termite soldier will put a lot of pressure on their mandibles, and they work to snap. So they'll snap their mandibles, and it'd be like flicking a bottle cap off of your table or your desk, and it goes flying off the other direction. Well, that's how these mandibles work. You can actually get on YouTube and somebody filmed this soldier in action. There was either an ant or maybe a termite from another colony or another species that came face to face with one of these soldiers. And this soldier snapped its mandibles and it happened so fast and so quick. I mean, it's really hard to see where that individual ended up flying off to. Here we have a termite. It's a drywood termite, and it's a species called Cryptotermes cavifrons. And it has this plug-like head, or it's a phragmatic head. So instead of actively snapping or pinching or crushing, it uses its head as like a shield to prevent entry into the nest from some other invading species. Here we have the conehead termite, Nasutotermes. This is a termite that's invasive to the United States, but it's native to the Caribbean islands. And they have this unusual head, this tipped head that looks like it has a big nose, and that cone-shaped forward projection that we see here, that's called a nosus. And from that nosus, these soldiers can secrete and spray defensive compounds. Now, these soldiers, they make up about 20% of their colony, and their noxious secretions can actually deter enemy ants and even large vertebrates, such as anteaters, for instance. You can also get on YouTube and see these soldiers in action, and it's a pretty effective form of defense. And then finally, here we have old workers that are equipped with exploding backpacks. That's the name of the scientific publication. And this is an example, really, of altruism to the next level. As these For this particular species mentioned in this publication, the workers, as they get older, they grow these sacks of toxic liquid that they can explode onto their enemies in an act of suicidal self-sacrifice. Pictured here is an image from that publication, and if you look closely, there's a couple of white arrows. And these arrows are pointing kind of to the thoracic region of these older workers. And you can faintly see there's like this bluish hue there. And those are the blue crystals that they use for this explosion defense. Now, self-destructive behavior is not necessarily uncommon among sterile worker castes of social insects, right? Think honeybees. We know that honeybees, they have barbed stingers. And when she decides to use her stinger and sting, that stinger remains attached to your skin. And with it comes the venom sac, and it rips catastrophically out of her abdomen. And so she stung not to defend herself, but she stung to defend the nest, because after she stings, she's going to fly off and die not long after losing her stinger. So that's something that we see here with some termite species, and researchers have found that this rupturing that we see with these older termite workers, it's evolved independently several times in a number of termite species. So this is an unusual example of defense because, like I mentioned, when we talk about termite defense, we think of the soldier caste, but this is an example where workers can help to defend the colonies too. So this is an image of that publication in Science Journal, and I'd like to share the abstract with you because it's so fascinating. It reads, by nature, defensive behavior is risky. In social insects, such behavior is more likely to occur in individuals whose potential for other tasks is diminished, aka they're older, they can't help out like they used to. And these researchers show that workers of this species develop an exceptional two-component suicidal apparatus consisting of copper-containing protein crystals stored in external pouches and internal salivary glands. During aggressive encounters, their bodies rupture and the crystals react with the salivary gland secretion to produce a toxic droplet. Both the amount of defensive substances and the readiness to explode increase with workers' age as their food collecting abilities decline. So the takeaway here is that termites have evolved some really unusual, really unique ways to protect their colonies, to ensure their survival. And if we've learned anything about the evolutionary arms race with insects, history has shown us time and time again that they are very good at adapting, developing better escape responses, more effective offenses, better weapons to fight with. The list goes on. All of these things help them to avoid elimination and live on to reproduce and ensure their genes get passed on to subsequent generations. They are a worthy opponent. Okay, fact number 4 here. Colonies can be founded in different ways. When we're talking about colony formation, we're talking about how they originate. The way that new colony gets started can vary in some cases. So we're going to discuss the different events that lead to termite colony formation and establishment. Now, during a swarming event, the king and queen pair up, they shed their wings, and they start to look for a suitable place to create and build their nest chamber. That mated queen will lay about a dozen or so eggs. She'll care for those eggs, and those first workers will start to forage on nearby cellulose. That could be little bits of wood, mulch, leaves, pine needles, whatever's kind of nearby, close to that nest chamber. As that queen continues to lay more eggs and more workers are produced, they expand the nest galleries and start to forage a little further away from the colony. New termite colonies, as we know, grow kind of slow. Maybe after the first year, that colony could contain around 50 individuals. Year two, maybe a little bit beyond 100 or two. And, generally speaking, it may take 5 years or maybe more for a colony to grow large enough to be able to produce swarmers of its own. And of course, all of those things are going to be influenced by a number of different variables and factors, but um, we do see that in some cases. And it's not uncommon for many people to think that swarming events are the only way that termites will create new colonies. But I want to discuss some of the other ways it might happen. For instance, if a portion of the termites in a colony gets separated from the rest of the nest, it's possible for subcolonies to form. This type of event is called a splitting event or a budding event. And let's think about some of the things that might lead to this outcome or what may cause a colony to split or bud. Some examples include, for instance, the secondary reproductives that go on to establish a new and separate colony, especially in cases where there's a large increase in the parent colony size. We know that for termites and then also some species of ants that have multiple queens, it's not uncommon that these splitting or budding events occur, and a portion of that colony ends up separating and establishing a new colony elsewhere. Physical destruction of the colony, this could happen potentially due to, a natural disaster like floods or droughts, or even something related to human interactions like construction, for instance, and then also the use of pesticides. Repellent pesticides specifically can cause that to happen, because ants and termites are very sensitive to repellent chemistries. So this is important for a PMP to understand, because we need to know how colonies form, how they are founded, how they get established, because that can help us understand how to eliminate them, and also how our own actions, for instance, using those repellent chemistries, can potentially lead to budding events. Fact number 5, termite kings and queens are long-lived. A few slides ago, I did have a bullet that mentioned that the primary reproductives of some termite species can survive 10 to 15 years, which is really crazy to think about for an insect living a decade or two. I know there were some ant queens in our colonies at the University of Florida that were 20 plus years old. These are insects that are outliving our cats and dogs, which is crazy. So how and why do termites live so long? That's a question that we're wanting to answer here. There's a few things that have led to scientific interest in termite aging studies. They exhibit some unusual physiological and ecological characteristics. They have long lifespans and very high reproductive outputs. So in the next few slides, we're going to look at the findings from this publication published in 2020. As you can see from this list, there are many things that contribute to this long lifespan that we see with termites. This is a table from a publication that summarized those ecological and physiological characteristics that help termite reproductives live longer. Ecological characteristics include nest defense, which of course decreases the risk of predation, thermal stress, desiccation, social immunity, which helps to alleviate risks with pathogen infections and reduces mortality, group foraging, which avoids the risk of starvation. And then also here we have hypoxic nests, which protects the reproductives from oxygen toxicity. At the bottom here, there are several physiological characteristics like oxidative stress resistance, which helps the overproduction of reactive oxygen, DNA repair, transposon defense, a lot of things essentially that are going on within the termite reproductive that helps to maintain homeostasis, which means maintaining stability that is critical for survival. Now, all that is said, I want to summarize that here. There is no one singular thing that we can point our finger to and say, this is why, or this is the secret magic behind the long lifespan and high reproductive output that we see from termite reproductives. We know that from these types of studies that it's most likely going to be a combination of ecological factors, nest factors, social factors, and even factors within the biology of the reproductives that contribute to this outcome. Now, historically, scientists have often observed the fascinating behaviors of ant and termite colonies for brief periods of time. No one really followed the demographic history of a whole colony throughout its entire life, which can last for decades until now. This is a blog post from the University of Florida that reads UF study sheds light on termite colony longevity and reproductive strategy over decades. Now, this is a fascinating research project done by UF entomologists that studied the long lifespan of specifically Formosan subterranean termite colonies. And they learned that each individual colony may attempt a different strategy to improve their chances of both survival and reproductive success. Let's talk a little bit about what they did. So this was a project led by Dr. Nanyao Su, and he initiated this work back in the late 1980s, where he monitored field colonies of Formosan subterranean termites. Now, the monitoring stopped in 2009, which produced 23 years worth of observations, resulting in over 15,000 data points. Tremendous amount of work, tremendous amount of data, and it required the scientists at UF a decade to actually comb through and analyze and make sense of these findings. The researchers at UF determined that the field colonies exhibited different survival strategies. In some cases, the colony lost their primary king and queen, but was able to maintain that reproductive output through long inbreeding cycles with secondary reproductives. Other cases, the colonies failed to produce replacement reproductives. And if you don't have any backup reproductives or secondary reproductives, there's nobody there to continue to lay eggs or grow the colony, ultimately leading to colony death. And then lastly, they also observed colonies that would produce thousands of replacement kings and queens and allowed the nest to survive and thrive for decades. Now, pictured here is Dr. Thomas Shavank. In the blog post, he compared the findings to the changes that can happen in a big city over time. Think Miami, think Dallas, right? The Miami of 2025 is dramatically different than the Miami of 1925. Infrastructure, demographics, economic productivity. And so he drew the parallel and said that for termites, it's similar, of course, but on a different scale. You have generational turnover. And so just like our cities, termite colonies are also resilient. They do change over time, over generations, and just as each city is unique, each termite colony is unique as well. So this data set revealed that each colony trajectory is different. It's unique year after year over the course of its life. And this study showed that a mature Formosan subterranean termite colony has many different options and various paths that it could take to optimize its reproductive output. It can switch from one strategy to another over decades to survive. So the bottom line here is that there really is no textbook answer for what will happen or what we can expect to happen to a termite colony over time. There's a number of variables that are going to affect that, and it's not really possible to predict. And so now that we know that information, we can take that into account for future pest control solutions. Moving on to fact number 6 here, humans are good at adding complexities and challenges that affect termite management, something that most of you are all very well aware of. And of course, the nature of construction being one of the big ones. There are places around the structure that are going to be particularly susceptible to termite attack and activity. And we're going to talk about some of those places and some examples of things that can make an already difficult task that much more challenging. So, starting with construction complications, one example being form boards. Anytime form boards are left in place, this is an example of direct wood to soil food source, could potentially invite or direct termite activity towards the structure. Cold joints, post-construction new slab pours like what we see here, this patio or this addition getting put in on this house, one example of many, right? They can be difficult to see and usually require indoor drilling to treat. Some buildings have serious foundation cracks, openings that can provide adequate entry for foragers. We know this is an example that is really difficult to see. One that is challenging to determine the exact extent of. It could be hidden within the walls or covered by the flooring or other hidden elements of the structure. Right? We know termites are cryptic by nature, and so these hidden flaws make it very difficult. This is a picture of a planter right up against the house. We see this one here is made of brick, but it could be made of stone, sometimes wood. The issue here is pretty obvious. You're bringing soil above grade, moisture, very little accessibility, visibility to those spaces, and can be quite difficult to treat sometimes too. We have foundation penetrations, right? Entry points for termites, bath traps, plumbing penetrations, electrical penetrations. Additionally, these spaces around the structure can offer termites moisture and protection, which can exacerbate the problem. Picture of a pier and beam foundation, right? Those can be difficult. They usually have low clearance, which makes it hard to access. Sometimes you come face to face with various dangers as well. Wildlife that can be hanging out in these areas, snakes, feral cats, possums. I'm sure many of you have horror stories of the things that you've encountered in these areas. It's dark, it's enclosed, be very difficult and hard to navigate. And not only that, but also, especially with really old homes, it presents the possibility of being exposed to previous termite treatments with those older chemistries that we know can persist in the environment for quite some time. And then foam, of course, many parts of the country, foam insulation is a major pain. The concern, of course, with foam is that it makes it very, very difficult for PMPs to do a thorough inspection for termites. Mission impossible, where we are unable to put our eyes on those key structural timbers that we need to look at, that we need to inspect for termite activity. And if you can't conduct a proper inspection because it's impaired by the use of foam, customers could potentially develop a termite problem right under their feet. So, how is this information helpful for a PMP? We know that termites are complex. We know that they are cryptic organisms. There's a number of things that people can do in the environments around the structure in the home that can encourage or worsen termite problems and especially complicate our termite management strategies. Coming along to fact number 7, termites interact with other animals other than to be their food, because another beneficial fact of termites is the fact that they are an important food source for a lot of organisms we care about: birds, bats, frogs, reptiles, amphibians. They're feeding on termites. So that's another benefit to add to some of the benefits we discussed a few slides ago, like they reduce compaction of the soil and also decompose wood and organic materials. So let's talk a little bit about their interactions with other animals. First, we'll discuss ants. Now, ants and termites are extremely abundant. In a publication that came out in 2022, they conservatively estimated that there is over 20 quadrillion ants on Earth. We can't really conceptualize a number of that magnitude, but that's 20 with 15 zeros behind it. And the biomass and global count of termites is equally as enormous. So their populations, of course, are going to overlap. Ants and termite populations are going to affect each other as well as the environments where they are found. They are both abundant, successful, and very dominating organisms. They exist above ground and below ground. And while termite predation does occur, mostly opportunistically, there's an indication that this interaction can affect other important processes within the ecosystem, like nutrient recycling, nutrient availability, plant growth, distribution, and more. Now, beyond ants, we have some termitophiles to talk about too. These are critters that live in close association with termites and even within their nests. Active termite nests commonly house various species of rove beetles, for instance, and they are sometimes described as highly specialized, oftentimes socially parasitic organisms. And although these species are commonly found inside the nests, information concerning their behavior is still quite scarce. But what we do know is that they benefit from this interaction that they have with termites by exploiting the resources in the nest. So here's a picture at the top of what a termite looks like, and at the bottom, B and C, we have a bird's eye view of that beetle, and then a side view of that beetle too. It has this physogastric abdomen that it kind of like folds over on itself, so you can see it has a similar appearance to the worker termite. Now, this is a beetle species that evolved to look like termites, and so they have this body form that enables them to mimic termites and socially integrate into their colonies. But one thing we know about termites is that they have poor vision. They don't rely on good sight in order to sense the world around them. They more so depend on pheromones, right? So there's evidence that these beetles have also found a way to also chemically blend in. There's also some beetle species that do this in ant colonies as well. They mimic the ant body shape. They produce similar chemicals and pheromones to convince the ants to feed them and to take care of them. So rove beetles, for instance, they're going to prey on the young, steal food, all while masquerading as a harmless nestmate. Moving on to another weird relationship that I'll briefly mention here. Insects and fungi, of course, they share a long history associated in various habitats, both playing the ecological role of the decomposition of wood. And fungal mimicry of termite eggs specifically is one of the more unusual evolutionary consequences of this termite fungus relationship. Some termite species will even harbor these fungal masses called fungal balls or termite balls along with their eggs in nursery chambers. These termite balls, which is a fungus, they require this sophisticated disguise to kind of blend in. They have this chemical camouflage, and so they can kind of blend in with the termite eggs. And the question here is why? Why would this happen? And what is the benefit of the fungus to blend in with the nest? Well, this camouflage enables the fungus a competitor-free habitat within the termite nest. This is an example of mimicry, very, very unusual. And essentially, it's convincing the termites to care for them. Now, on the same vein of termites and fungus, there's an article published a few years ago by the National Science Foundation about the oldest fossil evidence of farming. Termites were farming fungus approximately 25 million years ago, and here we have fossil evidence from East Africa that revealed that termites actually farmed and grew fungus. They arranged them, they took care of them, they fed them bits of plant material, and in return, the fungus provided nutrition for the termites while also converting plant material into a more digestive form. What's especially interesting is that the researchers discovered that the fungus only grew when it was cultivated, and so these organisms truly depended on one another in order to survive. So how can we use this information, or how does this change our understanding of termites? Well, for one, what I can say here is that the researchers involved with these studies emphasize the intelligence of social insects, which is an attribute that we rarely afford, especially to the pests that we face. Coming along to fact number 9, termite casts are not predetermined. So this means that the queen is not laying a soldier-specific egg or a future queen's specific egg. So the question here is, how does it happen? Well, we know that termites are used social and they have a caste system. There's division of labor, as we talked about. And I think we all would agree that the termite casts have very obvious, very specialized morphologies based on those tasks, right? Again, the soldiers have those large pincers that they use to defend. And in the reproductives, the queen is a very large egg laying machine and she has that body shape to support those activities. So, what then triggers the development into these different castes or specialized castes? Well, castes within a termite colony, again, are going to comprise of reproductives, soldiers, and workers, and the caste composition can be altered depending on the circumstances within the nest. And the developmental pathway will be one of two options. We have a linear pathway that we see here, as well as a bifurcated pathway. And the queen, she's going to lay an egg, and from that egg hatches a larva. And each individual larva has the potential to develop into any caste. Each individual larva has the potential to be either fertile or non-fertile. So what is going to dictate that pathway? And again, real quick, castes that are divided into two main types, fertile versus non-fertile, fertile being reproductive, of course, non-fertile being non-reproductive. Those fertile termites, they're going to include primary reproductives, secondary reproductives, alates, and those non-fertile individuals are the workers and the soldiers. And typically it's going to be determined by several factors, right? There's environmental factors, there's nest factors, what they get exposed to post embryonic development, as well as sex and egg factors. This can include nutritional factors, seasonal factors, temperature, humidity, as well as pheromones and hormone levels within the nest that they get exposed to, that in many cases is driven by the queen. What's especially fascinating about these superorganisms is that colonies can change their inner conditions by individual actions of colony members. So, by understanding the basic biology and how termites behave and how they develop, we can better understand how to manage them. For instance, many of the IGRs that we use in pest management were inspired by insect hormonal signaling. We're using their own natural biological pathways against them. Coming along to fact number 10, it's tough to be a growing termite. We're not going to have too much sympathy though, but I do kind of want to go through the different phases of molting. It includes abdominal contractions, splitting of the back. There's a breech area on the exoskeleton that they'll pull out of from. There's separation of the head from the previous head capsule and the antennae and the legs. And then 4 is that tracheal separation. So here's a visual picture of each of those 4 phases happening. You see that dorsal breach area that's kind of opening on the old exoskeleton along the back, and this is where the insect is going to pull itself out of. It's going to separate from the antennae, the head capsule, the legs, and then lastly in image D, we see those long longitudinal trunks being pulled from the spherical openings. What does it feel like? Probably not very good. Abdominal cramps, you've got contractions, your body splits open, your skin peeling off. This is a little dramatic, but at the end of the day, this is a very vulnerable place for all insects to be, right? They depend on their chitin and their exoskeleton to protect them, right? That is their barrier to keep harmful pathogens away, protects them from potential injury. And so this is a very vulnerable place for them to be, and it's shown that nestmates will be present to help that individual who's molting. They'll groom and remove those tracheal tubes, help to get that old exoskeleton off of them. So they look out for one another. And this is an important process to understand ultimately because PMPs that are using baiting systems, for instance, in many cases, those contain active ingredients that are chitin synthesis inhibitors. Excellent group of active ingredients to control termites, and this is the very process that they affect, is this molting process. So it's important to understand. If you don't have chitin, you don't have that structural integrity of your exoskeleton. In many cases, of course, you're going to die. Fact number 11 here, some subterranean termites can live without soil contact. Now, of course, we know subterranean termites depend on the soil. They need those moist environments. In order to maintain those ideal conditions, they build these mud tubes or shelter tubes that protect them from light and heat and other harsh elements. Now, obviously, given the name, they live in these underground areas and forage in these fairly expansive interconnected galleries in the soil. But when they traverse particularly above the ground, we know that they bring that material, that mud tube material with them to build these tubes. And the soil serves as a critical source of moisture that protects the termites from desiccation. It shields them from predators and is used as a building material as well as for carton nests above the ground. They are organisms that are soft and squishy, and they're going to be very sensitive to changes in temperature and humidity. So they bring that soil with them. As you're probably well aware, Formosan subterranean termites are known to build these carton nests, and they're using excrement, chewed wood, soil, mixing it together to build these tubes. And so they, not just tubes, but the carton nest material. And we find them typically where you have moisture issues in the structure. That could be voids, attics, even crawl spaces. And the issue here with carton nests is that termites can live for years on the moisture found inside these carton nests and in these areas where they feed on the wood in these above ground portions of the structure. Also, we talked earlier about how colonies can split or they can bud. And if that occurs and the colony gets separated from its subterranean nest outside of the structure, then those termites inside can survive in these carton nests for quite a long time, continuing to feed and continuing to cause damage. So formosans in particular are extremely difficult to manage, and the carton nests that they build and construct can be an issue because they can survive airily in that formation. And then lastly, real quickly, we know that termites can eat other things outside of wood. Now, the ones that we're concerned about for structural pest management, they're the ones feeding on wood, but it's good to know that there are different types of termites. Typically, historically, termites are going to be split into two distinct categories, higher termites and lower termites. And what that termite species is eating and how that food gets digested more specifically is going to dictate what category they get put into. Lower termites, they're digesting wood with the help of those gut microbes, the protozoa, symbionts, whereas the higher termites, they have lost that protozoa, but they have developed complex relationships and associations with fungi and bacteria. And as we know, we like to put things into boxes, and that's true for most of the CEUs you've probably sat in. Like, we like to put things into groups and boxes, but not everything is going to fit. And that's certainly true here too, because scientists have found some species that actually interface with both of these categories. So, generally speaking, higher termites can produce their own cellulase enzymes, but generally rely on those symbionts, and they lost the key protozoa that the lower termites have and depend on to digest wood. Now, lower termites, again, they're feeding mostly on wood. Those higher termites, what are they feeding on? Things like animal dung, organic matter, leaves, roots, etc. And as we learned from a few slides ago, there's some that actually farm fungus in their nests that they use for nutrition as well. So the important thing to note here is that not all species of termites, even here in the United States, are going to be structurally important. Some, for instance, agricultural termites, right? They feed on weeds and grasses, especially in pastures and hay fields. They create that mud tube material over the grass. They can often get mistaken for being a structurally important species because they do look similar, but they are indeed different and non-structurally important. If you were to look at, for instance, the soldier cast of the agricultural termite and compare it to, for instance, the Formosan subterranean termite soldier, they would look very similar, but the agricultural termite soldier would have a little bump on their mandibles. So there's things that we can look at to help differentiate the two. I've made it to the 55-minute mark, and I just want to say thank you for your time and your attention today. And hopefully, by understanding some of this basic biology that we discussed today and those not so basic behaviors, these are things that you can remember, that you can keep in your back pocket and help make you better as a pest management professional. Thank you again.